online on YouTube?
1 minute. 1 minute. Wait.
Have to mute.
And yeah, we are on.
Okay, so thank you very much. So welcome everybody to our next talk in our course on biochemistry and molecular biology of plants. Today we have the honor to have another international researcher to join us for a seminar, Dr. Ana Maria Fortes from Portugal. Ana, as is a tradition here in our course, I briefly introduce some aspects of our course, but Leandro is the person responsible to introduce the speakers. So I will remind the students, people outside, let's say, on YouTube, that you are free to leave your questions in either in English or Portuguese. We kind of understand Spanish, although Magda is not here today, but we can understand Spanish and Italian more or less. So we have a few possibilities here as well. And for students here, I will briefly switch to Portuguese just for a very brief reminder to them. Then I will switch back to English and give the words to Leandro to introduce the speaker of today. Pessoal, quem quiser fique à vontade para deixar as perguntas em português no chat, que depois, obviamente, a professora Ana vai ler, vai responder, a gente faz a interação de sempre, tá certo? Mas pela presença do professor Minocha, a palestra vai ser em inglês, tudo bem? So, switching back to English, I thank you once again, Ana, for accepting our invitation for this talk, and I will give the words to Leandro to introduce you. Thank you very much.
Thank you.
Thank you, Igor. Thank you, Professor Minocha, to be here. Thank you, Professor Ana, to be here. It's an honor. Briefly, because your professional career is very exciting, but briefly, Ana Margarida Fortes obtained her degree in applied plant biology in 1996 and PhD in plant biotechnology in 2003. Ana is currently an assistant professor at Science Faculty of Lisbon University and leader of Fruit Functional Genomics and Biotechnology Group. Her research interests have been focused on fruit ripening defense. The major goals of her research are the establishment of putative models of fruit ripening and fruit pathogen interaction based on omics data to be further validated by functional analysis, in particular gene editing. She is the co-PI and participant of several other national projects and on grapevine. It's very wonderful to study grapevine because we love vine. And thank you again. The class, the students are yours. Feel free to start.
Okay, thank you, Leandro and Igor, once more for this kind of invitation. Right now I am going to put the presentation. So I no longer see you, but if you have any doubts or if I'm going too far, you can always say something and we can take it from there, because I'm not familiar with all the concepts that you have already apprehended. So today I'm going to talk about molecular, hormonal, and metabolic events that happen in fruits of grapevine, in grapes, under infection with Botrytis cinerea. And we are going to compare what happens in a highly susceptible cultivar and compare it with the resistant one. Just to introduce you the problematic, so Botrytis cinerea is a fungus that infects a lot of plant species. We can have infections of tomato, of orange, many fruit species. But in grapevine, it's one of the fungus that is mostly responsible for economic losses. And in this respect, in Portugal, we have a cultivar, which is called Trincadeira, that can produce amazing wines. Are you still hearing me? All okay?
Yes, yes. Okay.
Okay.
I cannot see your screen. I can hear you, but I see myself on the big screen and not your screen.
Well, maybe it's something that you chose to. Did you pin yourself on the screen, Professor Minosha?
I don't know.
You know, if you put your cursor in your screen, there will be a pin. If you pin yourself, because then you will fix that screen to you, and maybe you can disable that option, because we can see Anna's presentation normally.
Okay, I think I got it. Thank you very much.
You're welcome.
Okay, so I continue, and so this. Actually, the cluster is very compact. And since it is very compact, it can kind of form like this microclimate in between the berries that are very appropriate for these fungus infection. So in order to gather knowledge on these plant pathogen interaction, we perform some infections in experimental vineyard in Lisbon, in an institute next to my university, Instituto Superior de Agronomia. And we have studied how these grapes responded to the infection, taking into account many analyses. Namely, we started, and this paper has been published some years ago, we started with the transcriptome and the metabolome analysis. I suspect that you are familiar with what is transcriptome and metabolome. So basically, transcriptome corresponds to the analysis of all the transcripts that are expressed in a given tissue. And for the metabolome, the same, but concerns the metabolites. And we have integrated this information, and we could see that actually the grapes, they respond with a defensive program involving hormonal regulation. I will come back to this later on. But this defensive program that they activate under infection is not efficient enough, and the berries develop high symptoms of infection. So we were wondering how this would happen in a resistant or tolerant cultivar. We knew that this defensive program was activated, involving, for instance, jasmonates, as just to pick up one of the hormones involved in this process. But we were wondering what would happen in a tolerant cultivar. So what we did was to infect the grape clusters at this developmental stage, which is called EL29. This is a classification that we use in grapevine development, but basically, let's think that the berries here have the size of a pea, okay? And then we have collected more or less after 2 weeks, when the benches are already hanging down, and they start to have active cell division. And it was very interesting because, and surprising for us when we started this study, because we saw in the literature that green fruits, no matter which kind, can be tomato or whatever, they are kind of resistant towards Botrytis infection. But actually, we have obtained, as you can see here, for Trincadera, not for Syrah and for many other cultivars. This is not what happens. We could see already at the green stage berries with heavy symptoms of infection. You can already see here some sporulation of the fungus. So it can be also that this Trincadera is extremely and remarkably susceptible. And if you can observe this figure here, you can see that the cluster is already highly compact comparing to this Syrah. And this may be the reason why it is so susceptible. And what we have done, we have performed several analyses, namely in this paper that we have published 2 years ago, and I will come back later on to all these analyses that were done. But just to introduce you, what we could see from this hormonal metabolism is that it looks like this Syrah is tolerant because it presents high levels before infection of hormones such as jasmonates and salicylic acid, among others. So it seems that this tolerance is mostly based on pre-activated defenses. So keep this in mind, and now we are going to explore this a bit further. So basically what we did in this work, we infected, and also we did mock treated, of course, control, and the berries were collected at 3 developmental stages: the green, the veraison. Veraison corresponds to the one set of ripening, and you can easily recognize it is when the berries start to accumulate anthocyanins. And the harvest. So what we could see already from visual inspection, you already have heavy symptoms of infection in the green Trincadera. I hope you are seeing my arrow. Are you the mouse?
Yes, yes.
Okay. So here you can see already heavy symptoms of infection that continue till harvest, and harvest it gets really damaged. And in Syrah, at the green, almost nothing. There was some dehydration of the berries. They start to appear a bit in veraison and a bit more at harvest, but nothing when we compare these two is totally different. And we collected these for hormonal profiling, thyroid PCR analysis, and metabolomics, and more recently, we already have the RNA sequencing data that I will show you. So, the first thing, we knew from the visual inspection that the green berries of Trincadera were, and I would like that you focus, though I'm going to present results throughout the ripening, I would like you to focus more on this initial stage, the green stage, because it's actually the one where you have every symptoms of infection in Trincadera, and you have very low symptoms of infection in Syrah. And this we could confirm by qPCR using parameters specific to the fungal genomic DNA. And note that here is much higher than in 32. And in harvest, you already have some infection, but nothing comparing to Trincadera. And now the hormonal profiling, we did this in collaboration with a German group. And we use HPLC MS platform. I don't know if you are familiar with this platform, but if you want to know more, you can always put on the chat and I will explain you. Basically, you have the separation of the molecules according to they are first separated in liquid chromatography, and then they are separated on the basis of their mass, in the mass spectrometry. And what we can see here, we have the quantification of salicylic acid, and we did more genes, but anyhow, you have here the gene expression regarding genes coding for ADS1 and PAD4 and PR1 that are involved in the signaling and response associated with salicylic acid. And what we could see is that we have higher basal levels, as I told you before, in sera. And then what we see is that under infection, the sera doesn't change much their levels at this stage, but Trincadeira it does. So she actually activated defenses, but they are delayed. They are activated under infection. So probably the susceptibility comes from this delayed activation of defenses. And we could integrate this information, for instance, with ADS1 and PAD4, where you can see that here at EL32, green berries, higher basal levels of expression of this gene, comparing with Trincadeira, and then the activation that I told you under infection. It was a bit what we have seen here in PR1, that is related with the response activated by salicylic acid, but we didn't notice differential expression here, but we notice in ADS1. So this what accounts for salicylic acid. And then in this hormonome, we also included the precursor of jasmonic acid, LPDA, and also the conjugated form of jasmonic acid that has been referred as one of the most important molecules in a signaling associated and Trincadeira activated under infection. I will not go into details about this, but we could also integrate this information with genes involved on the synthesis of jasmonic acids, such as alene oxide synthase and OPR1. And we could actually have very nice integration of gene expression and metabolic profiling in this AL32, and the same doesn't hold true for the other developmental stages. But one thing is very interesting. For this transcription factor, this MYC2, we have, again, the same thing. And we know from the literature that mutants of this transcription factor have been reported to present higher susceptibility to Botrytis, indicating that this MYC2 may be a positive regulator of defense. So we continue, and what we can see in auxins, it's funny because auxins, they have been very much connected with the process of growth and development, but they are not like these classical hormones involved or being described as involved in defense, namely against necotrophs as Botrytis cinerea. But recent data obtained by other researchers that you can search on the internet starts to refer that actually auxins may play a role in defense, eventually in interaction with jasmonic acid, which is a classical hormone involved in defense against Botrytis cinerea. And again, the same thing as I told you, just focus on these green berries, and we could also integrate some of this information with genes coding for enzymes involved in auxins metabolism. So to wrap up this part, we also quantified this abscisic acid. This abscisic acid is more related with susceptibility, but this is a bit more complex, because actually we know that during grape ripening there is a peak of content in abscisic acid at various zones, which was confirmed by us during normal grape ripening. But what this can be, what we can think about this is that eventually abscisic acid is involved in susceptibility because it promotes the ripening, it accelerates the ripening. And the more you accelerate the ripening, the more the grapes get susceptible towards Botrytis cinerea infection. So it may be that actually this abscisic acid is at a certain extent produced also by the fungus, or it can induce the synthesis in the host in the plant in order to accelerate fruit ripening and therefore fruit susceptibility. What we know is that it can be, it is for sure at a certain degree. Well, for sure, we didn't quantify the enzyme activity, but it indicates that the gene expression in the plant, that the gene coding for this enzyme involved in abscisic acid synthesis is actually activated in Trincadeira under infection. So it seems that this increase in the abscisic acid may be due to this transcriptional reprogramming that occurs in the grape itself. I hope I'm not being very confusing you, so I would like first to wrap up this part for you to keep in mind what is going on regarding hormonal metabolism before I go any further. So basically, prior to infection, we have active defense in sera. You see already all these bunch of genes and hormones activated in red, and in Trincadeira they are still not activated. But when you pass to the stage of infection, you see that there are a few changes regarding sera, basically keeps the same levels, and in Trincadeira it's all activated. So again, regarding hormonal metabolism, that you know that is deeply involved not only in developmental processes but also in defense processes, you can see that all these strategies are activated in Trincadeira but delayed comparing to sera. So some ideas for you to keep in mind. It's that these signaling pathways of the salicylic acid and jasmonates have been reported to be either antagonistic or synergistic. And what we can also consider here, and it's also an important idea to keep in mind, is that it seems that the defensive response doesn't depend on one hormone in particular, but maybe the balance among the hormones may lead to resistance or tolerance. Again, auxins are hormones with emerging roles in plant defense. Abscisic acid is involved in the regulation of the onset of ripening and possibly in susceptibility. Here we cannot be sure. We would have to do some functional analysis, develop mutants in a grape, which is not easy, in order to really ascertain the role played by abscisic acid in defense. But as a take-home message for you so far is that here you have two different genotypes from the same plant species, and you can see that they have different hormonal metabolism. So when you investigate these interspecies genetic variation of hormone networks, it may eventually provide you insights into possible manipulation in order to obtain grapevines with higher tolerance towards Botrytis cinerea. Everything okay so far?
Yes. Yes.
Okay. So, as I told you, we have investigated the hormonal metabolism, but we also did the metabolomics and the transcriptomics. I will start with the metabolomics because it was also a big amount of data that was generated in collaboration also with a German group that I will talk at the end. And what you can see here, so we could identify many compounds from different classes, secondary metabolism, but also primary metabolism, namely fatty acids, such as this one, this hexacosanoic acid. And we also quantified some volatiles. And what is the first thing that you see here? Look, when you compare sera infected and control, nothing changes. Not a single metabolite changes. So she's just there with their pre-activated defenses, and it's enough. On the other hand, this trincadera, it changes a lot. And where you compare control sera and trincadera, here you see a lot of higher levels in sera of certain compounds that can be involved in defense. And when you compare infected sera and trincadera, it seems that she kept the defenses still after infection. Another representation is here, where you can see that basically very few things change between sera and infected, and they were not statistically significant. And you can also compare that there is higher content of certain metabolites following infection. So, as we had for the hormones, we also could see here that high basal content in fatty acids such as hexacosanoic acid, alpha tocopherol, which is an antioxidant, epigalactocatechin, which is a secondary metabolite that has also been involved as described in defense, hexanal, which is also volatile that has been described as involved in defense. And there's also this terpenoid or ursoleic acid. They were all in higher content in sera. So again, our indication is that the defense in sera is due to a pre-activated metabolism. And then more recently, we have analyzed the RNA-seq data. When I talk about the RNA-seq, is sequencing of RNA. And this technology, the first paper we published, we did microarrays. With microarrays, we only had access to the gene expression of the plant. But with this technology, it has the advantage that you basically sequence all the transcripts that are there, and you will see that you have the majority of the transcripts are for sure from the plant, but you will also have access to the transcripts from the fungus. And this was quite an advantage, and we have some surprising results that I will talk about later on. I just want you to keep in mind that when we compare, so you do this RNA sequencing, and you have a huge amount, thousands of genes expressed, and then you can do this enrichment analysis according to their functional category. And here you have many functional categories such starch catabolism, stilbenoid biosynthesis, jasmonate biosynthesis. You have a lot of categories that were enriched in a certain comparison. But what was funny is that no category was enriched when you compare infected samples versus control in sera. And we only found in the upregulated, in the downregulated, we only found 3 categories. So she doesn't seem to be very affected by the infection. But look at Trincadeira now. Basically, she activates everything. We cannot say that she's susceptible toward lack of activation of defenses. It's just that it gets there very late. So it's basically she activates hormonal metabolism, jasmonate in particular, a lot of the transcription factors that have been connected with the defense, secondary metabolism, sugar metabolism, because of course they need energy to be directed toward defense, towards the synthesis of secondary compounds that are involved in defense. And basically, she still gets infected. And then we looked, as I told you, here you have the multivariate analysis of these transcriptomics that we perform. And here you can see that the transcriptome of sera control versus infected is pretty similar as expected. And here you can see there are many differences between control and infected. But what was very interesting is that we thought, okay, so now we're going to take a look at the gene expression in the fungus to see what's going on in there. And here, it was, I was not really surprised. I didn't know what to expect, but it might surprise you. Where do you think that there will be more transcription reprogramming of the fungus? I mean, more activated metabolism in sera or in Trincadeira? I'm not sure I will listen to an answer, but anyhow, just to make you think about it before I say it. Excuse me. So, are you still listening to me?
Yes.
Yes. Okay. So, actually, it's in sera. It's in sera. We also looked at the functional categories, and we have much more genes expressed in sera than in Trincadeira. She activates cell division, she activates carbohydrate metabolism. The fungus activates here cell wall organization. A lot of stuff is activated in sera. So, actually, what we could see that a lot of proteins related with resistance and protein kinases that are known to be involved in defense are constitutively enriched in sera and triggered in Trincadeira by infection. So here I'm talking about the plant. I will go back to the fungus in a short while. The same about all these functional categories, ethylene biosynthesis, transcription factors, and so on, as well as secondary metabolism. But what we could see is that in the fungus, it's the opposite. So more activation of the fungus is noticed in sera, including many enzymes involved in cell wall organization, in metabolic, carbohydrate and fatty acid metabolisms. So we thought maybe when the fungus is already happy after a successful infection, why the hell you would keep virulence-associated mechanism if you already beat the defenses of the plant? So it may be, and this is something that we would like to analyze in the future, it may be that Trincadeira, before being infected, also shows in the fungus this high activation, this high virulent metabolism. But once it gets infected, these virulence-associated mechanisms are kind of down regulated. So you can see here like an image. So what we have? We have in sera, in the sera interaction, we have 653 Botrytis genes expressed in Planta, whereas we have 1/3 in Trincadeira, about 1/3. But the opposite is what we see in sera, the plant. We have this ridiculous number, 22, of differential success genes upon infection, but we have this huge amount of genes activated upon infection in Trincadeira. So it's quite different that here the defense is, the variety is still trying to respond and activating a lot, and the fungus somehow already knows and is already being inside. He already knows that he made it. And here it's the opposite. The sera, eventually she knows that she has enough defenses, and he is struggling in order inside the cells and lead to the cell death as the necrotrophic pathogen that Botrytis cinerea is. So, concluding remarks, we have pre-activated defense. I hope I convince you of this in sera, which are likely to be responsible for its resilience against Botrytis cinerea attack. Successful defense in sera putatively induce wide activation of specific signal pathways in carbohydrate metabolism in Botrytis cinerea. And this study also put in evidence that the pathogen actually can have a certain plasticity in its transcriptome, with several genes related with virulence and fungal growth that are expressed during infection in these sera green berries, which might be important for early stages of infection. And the last remark is that these new putatively elements of virulence might be target for functional characterization and might be eventually used to develop new and efficient control strategies. What I mean is that normally we think that we can develop tolerance in the plant, but eventually we can also diminish the virulence in the pathogen by gathering knowledge on the mechanisms that lead to successful infection. So I would like to thank the people involved in this work from the University of Lisbon. We also had collaboration, a long-time collaboration with the institute in Logroño. In this institute in Lisbon, we performed the infections, and then we have these nice collaborations with two groups in Germany, with whom we perform the metabolic profiling. And I thank you for your attention, and I'm open to all the questions that you would like to ask. Thank you.
Thank you, Anna. Thank you very much for the nice presentation. As a big fan of Portuguese wine, it was very nice to follow your presentation. By the way, I've drunk. Although I have I can start with the question. I mean, most of the time, the Portuguese wines they do not have only one single grape variety. I think I never drank a Trincadeira 100%. Is there a reason specifically?
Yeah, well, when you make we have this. There is also this trend in Portugal that is a bit changing now to do like a blend in the wines. But more recently, we also have this trend of doing monovariety wines. The thing is that when you do monovariety wines with Trincadeira, they are rather expensive. And because you cannot do in certain years, it's a cultivar that it presents very regular ripening in between seasons. So in certain years, it gives rise to fantastic wines. In other years, not so much. So this may be the reason why they are less common. But actually, that bottle that I had in the presentation was from a monovariety wine made with Trincadeira.
Okay, perfect. Thank you. It was a question that I had in my mind for the last couple of weeks that I was discussing with my father, who's also a aficionado with wines. So I can open now the questions to people either on YouTube. If you want to ask something, please leave your questions, or here in our environment. If the students, if you want to open your mic, you can also speak in Portuguese with Ana, of course, if you prefer, or you can even write the question in the chat. So I can already start because the students, they need to warm up. So I can start with a few questions. Ana, I think it was very interesting to see that Shiraz has these defense pathways activated even without infection. But I was wondering, normally, well, I mean, normally it says that you have a trade-off between growth and development and the defense responses. That the plant, once it's attacked by pathogen, it switches off the growth and development and then activates the defense. Is this somehow observed, I mean, in Shiraz? I mean, if you compare the yield between Trincadeira and Shiraz, does Shiraz produce less because the defenses are activated constitutively, or you don't observe that?
No, not really. But I understand what you say. Normally, there is this trade-off, and I kind of spoke a bit like that. When, for instance, you have this activation of the sugar metabolism, there is a downregulation of photosynthesis, and it gets sugars to be hydrolyzed and to be used for the synthesis of the secondary compounds. What I think is that for some reason, the secondary compounds that are produced in these all this metabolism is against Botrytis cinerea. But Syrah can be infected with other fungus. Okay, so I think it's there is no grape variety that is Vitis vinifera species that is a resistance to Botrytis cinerea or powdery mildew or downy mildew, which are also other fungi that are really damaging the production of grapes. So this is how I see it. That is a very specific metabolism that contributes to these tolerance. Actually, right now we just published the analysis of grapes infected with powdery mildew, and we are analyzing what is common and what is different. But we have some drawbacks here. The problem is that the cultivars are different, but this you cannot solve, because if they don't get affected, that's the only way you can compare. So it also has limitations. So basically, to wrap up and respond to your question, I would say that there is indeed some kind of trade-off, but I think the main reason is the kind of metabolites that are produced and the kind of the gene expression that is activated, like a specific program to defend against these fungus.
Okay, and what? Yeah.
I didn't say that.
I think so.
Oh, Igor, I think, Anna, if you prefer, you can stop sharing now. Click on the...
Okay.
Okay.
I thought I heard somebody
Yeah, this is better.
asking.
Okay.
That's much better. So Anna, can I ask you a question?
Sure.
When I was looking at your metabolome analysis and all that, I'm assuming that the plant material you're using is infected visibly. If it is,
yes,
then how do you distinguish between metabolites that are being produced by the fungus due to infection, not if the fungus was growing, let's assume, in a culture or whatever other way you can grow it?
Actually, that's a very good question. For certain cases, we cannot be sure. But what we can say is that when you collect the grapes, you have like 95% of the material is from the grape. Yeah. And like 5% is from the fungus. At this stage, at this green stage, if you do it at harvest stage, it will be a bit different. So already we think that the major contribution should come from the grape. But what we could see is that also the reeds, this was our first idea on the first work, but now we confirmed that also the reeds, when you compare the reeds of grapevine and the reeds of the fungus is much less. And actually it was funny because it was around like 5% of the reeds were with the fungus. So basically, I would say
I think you are frozen.
Yeah.
What?
Yeah.
Your voice got frozen. So
yeah.
Maybe...
I would say that the major contribution comes from grapevine, and in certain cases, we also integrated the information from gene expression with grapevine with the metabolites, such was the case of the hormones, and we could see good integration. But we always have the possibility that the pathogen is producing or contributing to the production of certain compounds. But that's also interesting because it can be maybe really related with is trying to activate some susceptibility.
So were you able to see any polyamines in the metabolites or no?
We didn't use these protocols. We didn't use these targeted for polyamines. We could not detect. But I have good news for you. We could see that in Trincadeira there was activation of polyamine metabolism. If I remember well, there was some arginine decarboxylase being activated out there under infection. Yeah.
Wonderful.
Yeah.
Okay, Anna, there's a question on YouTube. Before I read the question, just to say that Professor Eni Flo is saying hello to both you and Professor Minosha. She's following us. So thank you, Eni, for following us, and she's saying her regards to you.
Thank you very much. Obrigada.
And our friend Douglas Dominguez, he congratulates you for the nice presentations. He said that he's also a fan of varietal differences in fruit development because it's a colleague who works on coffee. So I think he's familiar with that. And he asks you whether, besides the resistance to the fungus, if the choice for Shiha also has something, or there's another aspect to select Shiha, and if it would be the best genetic background to be compared to Trincadeira.
Well, what we knew, and for here we have like collaboration with the plant pathologist, is that Syrah was strongly tolerant towards infection against Botrytis cinerea. This is what they have observed in the field. And I can tell you that normally we perform the infections. So it's already a nice environment for the fungus. But normally, natural infections do not occur in Syrah. So we were sure that this would be a nice model. But we are also dependent on the grapevines that are on the field. And if you compare Trincadeira with Syrah, you have to make sure, or if you compare susceptible with resistant, you have to make sure that they are in the same terroir, grown under the same circumstances. You cannot put many more variables on this. So it was both factors that lead to this choice.
Okay, perfect. Let me see something else. I have also another question.
Me too.
I was wondering if this, I don't know if you can answer that, but I mean, if Shiha has this activated pathway, if somehow you can untangle and identify how this affects the organoleptic properties of the grape. I mean, because you show that there are activations of phenyl propanoids and all of those localized metabolites. So maybe this is something that contributes somehow to the properties of winemaking based on Shiha.
That is true, but since we focus our study on the green grapes, we don't have much idea how it would affect the wine in that sense. But actually, not from the fungus, because they produce these mycotoxins and they give rise to the off flavors. But when you think, for instance, to drought, the producers, they apply this drought, which is the stress, and that leads to the production of certain secondary metabolites. So basically, yes, I mean, it does play a role for sure. And that actually, I never thought about it. I'm just thinking about it right now, because this Botrytis cinerea, it's an opportunistic fungus. You always have this fungus in the vineyards, and it's just trying to get the right moment to infect. And actually, we could detect a few reads in the control in the Syrah berries. So the fungus is just trying to get some nice conditions. Whether the fact that this there actually activates what you're saying, this we don't know, but it would be funny to, I'm not sure how we would do it, but well, it would have to be with a strong fungicide treatment, not very sustainable, to make sure that it eliminates all the fungus there.
Okay, thank you. Leandro, you can go.
You can read firstly the question from Douglas, because it's a continue.
Okay, I didn't see that other question. So yeah. Douglas continues with another question, Anna. He said that he was wondering about the breeding story. In coffee, I usually try to compare varieties that have somehow similar breeding stories, but have contrastant resistance. So, I mean, because then I think what he means is that you compare closely related genetic backgrounds, but with the difference regarding your target process. In this case, the resistance to the fungus. So whether there would be a better control or a better comparison to Trincadeira based on genetic background, I guess.
Well, considering that this Syrah is originated from France and Trincadeira is originated from Portugal, I wouldn't expect, though I didn't make much research into it, that they are not, that there could be more easily related regarding breeding. But considering that what we wanted mostly was that comparing a highly tolerant with a highly susceptible, that was the reason also to choose this cinerea. And in the field, we didn't have any other variety growing that was so tolerant to cinerea, to Botrytis as cinerea. So that was the reason.
Regarding, I mean, regarding let's say, like, considering the whole world, I mean, is Shiraz something somehow specific in this regard? I mean, highly resistant, or there are other varieties, not limited to Portugal, but I mean, is this?
No, there are other tolerant varieties. I was trying to remember one in South Africa, but I don't remember the name. That was also very tolerant. I think it's Pinotage that is highly tolerant. I have read about it. Some varieties are also tolerant, and but I really don't remember the names. What I know is that we have more susceptible, but in Portugal, grown in Portugal, Trincadeira is by far the most susceptible. For these, you cannot get a better model. Even when we infect, for instance, our favorite cultivar, Touriga Nacional, you don't get these symptoms in the green grapes. This is clear.
Okay.
So I would say from the susceptible side, it was the best model. From the cinerea, probably we could get another model if they were grown in the field. Yeah.
Perfect. Thank you, Leandro.
My question is about the auxin. Is very interesting because it's not common to see papers about the auxin related to the infection. My question is about the roles of auxin in this case. It's about the effect on the cell wall because the infection about in this case, or
it can be,
or some start to expression of another genes like by transcription factor like ARFs, auxin response factor, or another one.
This we also had. This we also had. Well, we just have, regarding your question, we just have some ideas because we cannot know for sure unless you do functional characterization that you develop mutants or overexpress certain genes. But yes, we had a lot of auxin response factors being activated. I also think it may be connected with the cell wall because these necrotrophic pathogens, they really digest the cell wall. So I would say that it can be connected with that. And I also think that since auxins are so much involved in cell division, it can eventually be some trade-off between the growth, and like some mechanisms related with that. But we cannot be sure at this point about what is going on there. No.
And another question about the ethylene. Do you think that because in this case that ethylene or genes related on biosynthesis of ethylene is more expressed or activated or not?
No, we had also for ethylene you cannot quantify with this platform because it's volatile. So we didn't quantify ethylene, but we had a change in the gene expression involved in the ethylene. So I would strongly suspect that ethylene is in higher content for instance, in Trichoderma under infection. Yes.
Yeah. Thank you.
Okay. Some other questions? I think all of my questions. I have one last question. Anna, I don't know. I think you mentioned, but I was, typing another question maybe. Why? I mean, you show that Shiha has the defenses activated prior to the infection, but then you show that in the first two stages that you analyze, the level of infection is very low, but in the end, even Shiha has a little bit of I mean, at least like by qPCRing the fungus to identify what is the shift that happens in Shiha that in the beginning it's resistant, and then in the end, I mean, maybe because of the amount of sugar that is so, I don't know. I mean, is there any mechanism that you identify?
We didn't study in detail these time points, but I can show you is that there is decrease in certain hormones, and there is also the decrease in the expression of certain genes involved in hormonal metabolism that may eventually be involved in defense and are no longer so much expressed. But we don't have detailed data on that. We didn't do metabolomics, we didn't do transcriptomics. So I don't have a strong response on that, but I suspect that basically what should happen is that there is a decrease in these pre-activated defenses. That's what I would guess.
Perfect.
Actually, it's something that we already saw in Trincadeira. When you compare the green berries with rhizome berries, there is a decrease in the activation of defenses in Trincadeira. So I suspect, but this decrease is already in control. It's in defense under infection, but also in control. So I would suspect that you also see the same in the Syria. Normally, they decrease the defenses during development, and here it can be also because they are accumulating more sugars.
Yeah, I thought
So they don't have so much precursors for secondary metabolites for defense.
there's more food for the fungus as well.
Yeah. He's happy.
Okay, so students, if you have questions, please let me see. No more questions. We still have some time. I guess everybody's happy. So I would like to thank you, Anna, once again. It was super nice to have you here.
Thank you for the invitation.
Maybe
and whenever anybody comes to Portugal, you can always mail me, and we go for a glass of trincadeira.
I was about to say exactly this. Then you can take us to drink some Portuguese wine.
Igor, I think that Professor Mendonça want to talk. Professor, your mic is muted.
Yeah.
Oh, yes, I raised my hand that we will certainly come to Portugal.
You are invited. We are welcome.
For some pastelzinho de Belém and wine.
Port wine.
Well, it doesn't go together. Better some cheese.
Do you make wine in your own lab also?
No. We just go for drinking every now and then.
Okay, great. So I officially would like to thank Anna and everybody for your attendance, and to the students and everybody else. We see you next week. Thank you, Anna. Thank you, Minoshas. Thank you.
Okay, bye-bye. Thank you.
Anna, if you can stay
Bye,
for
everybody.
about 2 minutes,
Thank
would
you.
you be able to? Can you stick around
Yeah,
for
Professor.
2 minutes?
Of course.
Yeah.
So I will stop the transmission on YouTube. Now, see you guys.
So bye-bye.